The last talk for today. I have an honor to represent here the research made by wonderful Tasmanian myxomycetologist Sarah Lloyd, which did not come on this Congress, unfortunately. And our team, Spanish, German, Ukrainian team, was lucky to be involved in this study. That is why I can tell you about this research. So, Australia, as you already know, is the hotspot of unknown, still undescribed diversity of myxomycetes. And in February and March of this year, 2022, we made an expedition there with which could be impossible without kind help of our Australian colleagues. And we visited different regions of this country and was able to work with the collections, including collection of Sarah Lloyd and also collection which is deposited in Herbarium of Melbourne. We also met a lot of nice animals, and those guys you can see on my photographs, they all are endemic to Australia or even to Tasmania, as well as many myxomycetes do. And another nearly endemic group is this nice team of non-institutional scientists who work to discover new items in this local flora. And this foray was held in New South Wales, but the species I will tell you about was collected mainly in Tasmania and Victoria. And this is not a first species found to be endemic for this region. Together with Sarah Lloyd and Gabriel Moreno and Marta Schnittler, we described already 5 species of myxomycetes which seem to be endemic to Australia, including 2 species of Alvisia and 3 species of Tubifera. But these all were bright spored myxomycetes, and now I will tell you about a dark spored myxomycete. This species, pretty conspicuous, forming large colonies which emerge from the bright yellow plasmodium, was initially identified as Lamproderma, even with the fact that Lamproderma should not have a yellow plasmodium, only the white one. But nevertheless, you can see a typically lamprodermoid appearance, a shiny film-like peridium, spherical sporotheca, long stalk. So one another Lamproderma, this was the first idea. But then the closer look has shown very strange additional details, especially a petal-like opening of sporotheca and capillicium, which detaches from the columella and remains as a kind of a fur on the surface of peridial lobes, as it may happen in Diderma and some Didymia. Microscopically, we could find that capillicium is tubular, poorly branched, and contain small spindle-like vesicles, which are also rather characteristic for Didymium, Diderma, and genera like this. Also, spore ornamentation, which is, of course, pretty variable in dark-spored myxomycetes. Also, it is not lamprodermoid enough because of these strange groups of smaller and bigger warts, which are also very common in Didymium, for example. As for the SEM pictures, here comes our input to this study. you can see that Capillicium tubules are verrucose, pretty strange for Lamproderma. You can see that they merge to the peridium, forming these furrow-like ends, which are opened outside. And here, this is a look from outside, and this hole comes inside to the Capillicium tube. And of course, this didymoid ornamentation of spores. And if we look at dark spore genera using traditional concept of genera, in which every genus must have a kind of plan of the structure, specific structural plan, then we can easily see that our new species has its own plan of fruiting body, standing somewhere between Lamproderma and Diderma. From one hand, it doesn't contain any lime and is generally Lamprodermoid. From the other hand, Capillicium is attached to the peridium, and it is attached so firmly that when the sporotheca opens, it remains attached to the peridium and breaks out from the columella. It is similar to Diderma, but of course, you know that in Diderma, in most cases, the threads are detached from both sides. So if you put all the morphological data in one table and do a phonetic analysis, a PCA analysis, you will find that species of dark-spored myxomycetes form 2 or 3 clusters, lime-containing cluster and lime-less cluster, and our new taxon is somewhere in between. So this is not only intuition, this is also quantitatively described. As for molecules, we succeeded to receive, and in this part also Angela took part in this part of the study, we obtained 3 independently inherited genes with pretty normal SSU, with pretty normal COX-1, but with very strange EF1 alpha, which was so deviating that blast search in GenBank could not find any good analog except of some protist and some plant. So we did not include this gene to the final analysis, but I think it is probably correct because the model of the part of protein of the elongation factor, which I built based on this sequence from the new taxon, is absolutely identical to the same portion of the sequence of Physerum polycephalum, with a tiny difference in this area. Of course, spirals are mainly same everywhere because it is functional and conservative part, but also these loops, which are not that functional and that conservative, also coincide. This is a final phylogeny, which is mainly 3 gene, but for new species, it is 2 gene. Of course, the 3 gene tree also was built, and it just gave a somewhat longer branch. That's the only influence of the 3rd gene included. And here we can see Physeracea, Didymacea, Lamprodermatacea, and our new taxon is situated on the base of the lime-containing branch, having a lot of common with the lime-containing Physeralis, except for lime, actually. So this fits well to the phonetic data we have. So the conclusion was that we have a right to describe it as a new genus, which was called Tasmaniomyxa, and a single species of this genus, which was called Tasmaniomyxa umbilicata because of the umbilicus on the base of the sporotheca. Interestingly, this species seems to be endemic, but not endemic of Tasmania, but endemic of the coast of Tasman Sea. This area is called Tasman Sea because There are rather numerous findings in continental Australia, in the Victoria state, findings in Tasmania, and also one single collection known only from the internet, from the database. But since morphology of the species is so peculiar, we believe that we can identify it by the photograph. So we have three areas in which it can be found, but never else. And the probable reason is that in this area we have specific vegetation, including green spots eucalypts and yellow spots nothofagus, two types of forests which probably represent the habitat of this nicely described new taxon. So the general conclusion is that if you come to some distant area, you can find not only new species, but even new genus. And moreover, we can even expect that it will represent a new family, but of course, we will wait to a good, reliable multi-gen phylogeny to say anything about this. Many thanks.